Okay. Oh, it's a great pleasure for me to introduce a colleague of mine at the Faraday Institute, Dr. Alexander Massmann. He joined the Faraday in 2016. His background is in theology and theological ethics. His PhD was from Heidelberg University, and he did a postdoctoral research fellowship here at Cambridge University as well. And he joined the Faraday to work on a new project, which is in collaboration with Keith Fox, looking at the topic of human genome modification and human dignity. And he's going to explore some of those issues with us now. Thank you very much, Alexander.
So after Jonathan started out with, I think, the ethical section, he also talked about the Google Earth, where you can zoom out and look at the Earth on the grand scale, zoom into landscape. So he laid out a larger view of things, preservation of this biosphere as a whole, and I'm going to zoom in even a little deeper into particular questions in applied ethics, or practical ethics. So since 2015, a few research teams in China and the UK and in Sweden have modified the genome of human embryos. Work on viable human embryos was first published more recently, just this past March. And in the US, the National Academy of Sciences argues that regulations in this area should be relaxed. Novel genome editing techniques, notably CRISPR-Cas9, place efficient and precise genetic modifications within reach. And resonance in the media is remarkable. As just one example among many, CRISPR was featured in a recent cover story of the National Geographic magazine called "The Next Human: Taking Evolution into Our Own Hands." The report did not waste much time dwelling on fairly conventional research that would alleviate or cure specific diseases. It reported Nick Bostrom's view of improving humanity in a different sense. So Bostrom imagines that a woman might undergo IVF and then pick those embryos that have an above average predisposition for a high IQ, and if that child then does the same thing, and if this procedure is repeated throughout the generations, Bostrom envisions, then our descendants might look back at the 21st century as the end of the Dark Ages. The journalist of the National Geographic story was perhaps a little uneasy. Is an artificial genetic increase in IQ realistic? Is this reason for hope or for fear? Should we, in a good scientific spirit, keep an open mind? The journalist concluded this bold outlook of Bostrom's into the future with a deadpan question: what the point of such biomaneuvering might be. After all, we're also told that in just a few generations, even the most intelligent of our own descendants will be vastly outsmarted by intelligent computers anyway. So why bother modifying genes in the first place? Futuristic transhumanist scenarios are not unusual in reports about CRISPR. Of course, much uncertainty is involved here. By contrast, this talk is going to ask first what the technology allows for, currently and presumably in the near future. What purposes can the technology be used for realistically? And what is the legal situation? Finally, I'm going to ask what we should think of human genome modification in ethical terms, both philosophically and from the point of view of Christian ethics. So first off, what is CRISPR? And so this is a schematic illustration. In short, CRISPR-Cas9 is a pair of molecular scissors that allows researchers to define genetic locations at which DNA is precisely cut in microorganisms, in plants, animals, and in humans. This allows us to render genes dysfunctional, which can be helpful for research purposes. Researchers might knock out genes in mice to see what their function is by scrambling up the gene with scissors, so to speak. Yet in using CRISPR, we might at the same time also draw on the natural mechanisms with which the cell routinely repairs DNA. This way we could sneak in a new stretch of DNA that would then be incorporated at a specific location. This would allow us to replace an existing dysfunctional stretch of DNA with a new functional one, or we might even add a completely new piece of genetic information. Due to the new level of precision, such procedures are commonly called genome editing. The possibility of precise, targeted cuts is not entirely new. For about 15 years, researchers have developed other genome editing methods. For example, so-called TALENs. The difference to CRISPR is that these earlier methods require much greater effort and precision in defining the target site to be cut. CRISPR, on the other hand, is easier to produce and significantly cheaper. Nonetheless, TALENs have been useful in removing or helping to remove blood cancer from at least 2 baby girls in a London hospital. This is a clear improvement upon the earlier gene therapy methods. Before genome editing, so even before those TALENs, some children, for example, with a severe immunodeficiency, were given a treatment that added a crucial genetic sequence to their cells. Their condition improved, but several of them went on to develop leukemia. The new stretch of genetic material was essentially dumped into the cell with no control if and where it would be incorporated into a chromosome. So this was before genome editing. We did not have the chance to make such precise cuts. And the faulty genetic sequence would remain in place. However, many parts of the human genome are finely tuned, and the procedure may either have created a new oncogene, that is, a gene causing a tumor, or it may have disrupted a regulatory gene that suppressed an existing oncogene. By contrast, genome editing holds the promise that such problems can be avoided. To modify genes in the grown organism, typically in adolescents or adults, downstream in development rather than upstream, is so-called somatic genome modification. At that point in development, the modified cells will divide and multiply only in limited numbers. For better or for worse, the impact is narrowly circumscribed. The changes do not affect the sex cells or those cells from which sex cells arise. So egg cells and sperm cells would not be affected if you use CRISPR in a grown organism. And those sex cells, of course, have a significant impact on one's offspring. But if our somatic procedure does not affect the sex cells, then genetic changes will not be present in the next generation. This can be an advantage, as results are better foreseeable. The hope is that some diseases can be cured or alleviated that result from mutations in just a single gene. However, one problem in somatic modification is that cells with dysfunctional DNA may be so inaccessible in the body that we're unable to ferry CRISPR into those cells. You'll recall that practically every cell in the human body contains the entire genome. Not all cells draw on all parts of the genome, but if a stretch of DNA does play a role in a particular organ, it does so in all relevant cells individually. A genetic mutation can keep, for example, all relevant cells in the pancreas from producing insulin. If you want to cure diabetes with a genetic intervention, then you'd have to identify those cells where this error occurs and replace the genetic stretch there, if indeed it is the type of diabetes that is caused by genetic problems. But the question is also, can you identify enough of those cells and actually reach them? That is, with diabetes at least, a significant problem at the moment. In addition, so we might need to modify a very large number of cells. For example, in neurodegenerative diseases like Huntington's disease, a potential therapy would require that we modify a significant amount of those 85 billion brain cells tucked away in our skulls. So that's unrealistic. Well, we might, what is more realistic is the modification of white blood cells, for example. This is how leukemia and the immunodeficiency SCID have already been treated successfully. Modification of white blood cells might also confer immunity to HIV/AIDS. This, to my knowledge, hasn't been done, but there are very promising approaches. Since a large amount of blood also passes the liver, a small yet critical amount of liver cells may be accessible to therapeutic modification. Current clinical trials are carried out with the hope of curing patients from hemophilia. Further, various studies in mice have given patients hope that CRISPR may alleviate the symptoms of muscular dystrophy. So those are potential uses of somatic genome modification. On the legal side, these somatic interventions, if they're aimed at therapy, they're not different from medical surgery or medical drugs. Of course, the procedure needs to be safe to pass the regulatory hurdles, but the fact that we are modifying genes in somatic cells rather than, say, removing an appendix, that's not a legal issue. And also on the ethical side, it would be difficult to find an ethicist who says that genome modification is something fundamentally different from surgery or medical drugs. While genome modification has already been done in this way with older generation genome editing methods, CRISPR itself, which is so much featured in the headlines, CRISPR is not yet quite precise enough for that purpose. The rate of false modifications is assumed to decrease significantly, however, in a number of years. So this is somatic modification. The situation is different, however, if we plan to modify an embryo. For example, while the embryo consists of about 4 or 8 cells altogether. At that point, cells are not yet segregated into the somatic line and the germ line. Here a change would affect also the sex cells, so modifications would be passed on to the following generations. That might be something one might wish to in fact achieve. Something else, why one might want to do that is that those changes would really affect then all the cells in the entire body, and then you no longer have the problem that certain cells would be inaccessible. So you're basically doing it from scratch. But this also raises the question to what degree the effects of our actions may be unforeseeable. In the UK, laboratories can apply for a license to conduct research by modifying embryos, but legally, modified embryos cannot be implanted. And that's the case for most of the countries of the Western world. They cannot be implanted in the US either, and on the European continent they can't either. In the UK, these embryos must be discarded after 14 days. Apart from the distinction between somatic and germline interventions, the question of purpose is important. Standard discussions will always appeal to therapeutic uses. Evidently, the therapeutic use of somatic interventions is clearly constrained by accessibility and numbers. But this should not distract from real achievements in this area. On the other hand, it must be clear that the possibility to cure, for example, SCID, is not necessarily an argument for germline interventions. Strictly speaking, germline interventions would not cure, they would prevent disease. Persons having a certain genetic predisposition or symptoms will not themselves profit directly from germline interventions. In theory, potential germline interventions might benefit the offspring of those people. For this purpose, however, parents would need to opt for the artificial fertilization of an egg cell in a dish, and followed by the implantation of that embryo. So this would be in vitro fertilization, IVF. The parents could do so if one of the parents has a significant genetic predisposition for disease. The embryo would then be tested genetically, and if it is found to have the critical gene, the embryo would hypothetically undergo genetic modification. If parents go that far indeed, however, then the question is why they would not choose pre-implantation genetic diagnosis instead. Pre-implantation genetic diagnosis, that is PGD, another one of these technical terms that's often thrown around in this context. So in pre-implantation genetic diagnosis, one obtains egg cells from the woman, fertilizes them, and then screens the embryos to see which ones carry a certain genetic predisposition, typically for severe disease. And those embryos that show this mutation are then discarded, and only healthy embryos, so to speak, are implanted. This procedure is not without critics either, as embryos are indeed discarded. Yet the question is, what the added benefit of germline modification would be compared to PGD. PGD can be suitable for avoiding severe life-limiting conditions when the genetic predisposition is limited to a single gene. That is the case with conditions such as Huntington's disease, cystic fibrosis, muscular dystrophy, Lynch syndrome, and various others. Now, if a critical predisposition involves various genes with a cumulative effect, then chances for a successful pre-implantation genetic diagnosis are lower. It is less likely then that among a number of artificially fertilized embryos, a significant number free of harmful mutations remains for implantation. So you'd end up with fewer, so to speak, healthy embryos that might be implanted. Only a limited number of egg cells can be obtained from a woman to begin with, and the likelihood for any embryo to be implanted successfully is roughly a third, and that's why the question is, do you have enough healthy embryos to establish a pregnancy? Now, if the mother carries several harmful mutations, the number of egg cells entirely without mutations will be low. This means that predispositions for disease based on several mutations might create problems for PGD. Germline modification could be helpful in that case. Yet, even that is not a foregone conclusion. Some cases of cancer, for example, are caused by various genes, but the precise genetic patterns that do indeed contribute can differ considerably between individuals. Two separate families may each have a history of very similar kinds of cancer, but the contributing genes may overlap only very partially. So in certain cases we may know that there is a high likelihood of cancer, but we might not find out what genes exactly would require editing if the troublesome mutations differ clearly between families. At some point, we may be unlikely to reduce the likelihood of disease dramatically with any method. And the question is if the difference that either PGD or genome modification can make justifies an elaborate intervention. Moreover, many diseases are not only due to genetic predispositions, but also due to significant lifestyle choices. In that situation, classic approaches in cancer prevention and therapy might be preferable. Finally, suppose that both partners each carry a gene on its own, that on its own increases the likelihood of disease significantly. Again, the likelihood of a successful PGD decreases, but now we know what genes are involved, and the disease risk is high for each mutation. Such cases are rare, but this could indeed be a case for germline editing. On the other hand, even here, PGD is not entirely useless, as one might screen sperm and egg cells individually before one fertilizes the egg cell. And that might increase the number of embryos without disease mutations. So the upshot is that germline modification is simply not the golden bullet in preventing disease, although the method could be useful in very particular situations. It seems that we will not miss out on highly significant benefits at the moment if we do not rush to change legislation concerning the implantation of modified embryos. It could be helpful to watch developments unfold in somatic uses of CRISPR first. And the discussion that is worth having in the meantime is what uses of PGD are legitimate. So the UK regulatory body, the HFEA, has published a list of over 400 conditions that make regulatory approval of PGD likely. So you can access this on the web, and the list goes on and on. If you have these conditions, that's an indication that they might well approve of PGD. Among these conditions are severe conditions like Lesch-Nyhan syndrome or Tay-Sachs disease. But then again, there are conditions on that list with much less severe symptoms. Hemophilia might mean a lifestyle constraint, but can be handled. But even diabetes is on that list. To what extent people really make use of this opportunity, I cannot say. But to offer PGD for conditions that can be fairly well handled and for which there are many resources may seem a bit excessive. The problem at this point is that we might lose sight of the fact that you can have a fulfilling life even with certain impairments. Such a life might not conform to conventional standards of happiness, but surveys have repeatedly found that people with disability tend to feel very satisfied with their lives. There are several empirical studies, so it's not just a hypothesis generated out of one's worldview. I see the danger that people might go to ever greater lengths to ensure that a child conforms to conventional standards, but they might forget that at the end of the day, what makes life worth living is not our own efforts. This is at least a point conveyed in the Christian message of God's undeserved grace. Certainly, CRISPR has the potential to create great scientific and technological enthusiasm, which creates considerable technological momentum in our discourse in society. add to that economic opportunities in the biotech sector. In this dynamic mix, the question of medical applications plays a crucial role, of course. So there's great momentum, but my suggestion is that we stop to take a sober account of worthy goals and genuine opportunities. There's a possibility, however, that our goals and values in life might be shaped by the technological opportunities rather than the other way around. And now I'm coming to another issue. So this was a look at the therapeutic possibilities and questions. And another question are so-called enhancements. So this is an issue that comes up in germline modification or also perhaps somatic modifications. But there's an interesting comparison to PGD also. In PGD, we compare between different embryos with slightly different genomes, yet we cannot introduce new genetic combinations there in just doing PGD. By contrast, germline interventions give us the possibility, at least in theory, not only to correct pathological mutations, but even to introduce new predispositions for positive characteristics. We might not only engage in disease prevention, but add new features to the organism. This is the area of the enhancement discourse. Suggestions for genetic enhancements are, for example, improvements in memory capacity, longevity, or muscle power. First, we would have to find out what genes one would have to modify. So we would screen the genes of large numbers of people of the population and establish correlations between certain genes and particular characteristics. Genome-wide association studies. First, a look at enhancements from a more technical point of view. The data collected by UK Biobank, for example, has helped researchers describe 72 genes that contribute to academic achievement. The authors of this study state clearly that a large part of scholarly achievement is due to environmental factors rather than only natural predispositions. But they put about 20% of academic achievement down to the genes. Hypothetically, and there are similar studies, other studies have identified entire genetic networks, and others have figured out what genes may be involved in autism, for example. Hypothetically, parents might then compare the genome of an embryo with that of academic high achievers, and they might find that the academic prospects of their offspring could use some helping with germline modification. One technical difficulty here is that this study only indicates what genes correlate with a certain number of years spent learning at school. A genetic modification might increase the chances that children succeed in their A-levels. However, genes for a master's degree, a PhD, or even a Nobel Prize would be an entirely different matter. Further, children might of course just use discipline and diligence in order to make up for what they appear to lack in genetical endowment. There might also be trade-offs. We don't know. Might the child perhaps lose other emotional qualities or end up with scholarly talent, yes, but reduced practical skills? We heard in Professor McMahon's lecture, for example, that lots of brain phenomena are emergent processes, so it doesn't follow that if you maximize one component, then the overall result is equally maximized. It might also be a question of the relation between the different elements amongst each other, so these are very difficult questions to handle. Other enhancement possibilities might include modifying physical height, which tends to correlate with higher professional earnings. especially early in your career, if you're at the finishing school, at university, etc. There are interesting studies on that. Another classic issue in this discourse is a significant extension of the human lifespan, and this occupied Francis Bacon, whom we've also encountered in these lectures here, who unfortunately died of a cold, so for him extending the lifespan meant something else than for us. Strangely enough, transhumanist philosophers have made the point about enhancing the lifespan by pointing to so-called Methuselah mice. Those are mice that were indeed modified to live longer. But not only are they sterile. From the perspective of evolutionary biology, it also seems plausible that in mice there might be room to tweak longevity. But that does not mean that the same is true for humans. To begin with, if mice were simply able to breed longer, they would have a clear selective advantage in evolution. So you'd expect that trait to be passed on and to spread. If it is possible to extend the breeding age in mice, the typical evolutionary mechanisms have probably already done so. Now, might we perhaps create a mouse that goes through menopause and then lives significantly longer without breeding? Should a mouse strain in the wild turn out to live longer beyond breeding capacity, that would not increase the number of their offspring. So in the evolutionary scheme of things, they would not profit. That's not a trait that would be rewarded by natural selection. So if there are genetic ways to achieve that particular pattern of longevity, evolution is unlikely to reward them and ratchet them up, and in consequence, the mere fact that there are no mice in nature that go through menopause and continue living does not mean that such an effect would be impossible to achieve artificially. But how about humans? The situation is different here. In humans, evolution has already explored routes that imply longevity. We are one of the very few mammals in which females go through menopause and continue living. The key reason is that after menopause, women contribute to their offspring's Darwinian fitness by caring for their children and grandchildren. The longer, the better. So in humans, evolution has already rewarded, honed, and tweaked those traits associated with longevity. Just from this evolutionary point of view, it is not at all obvious that a significant genetic extension of human longevity is in the cards, even if that might turn out to be true for mice. So these would be hypothetical examples of enhancements via germline editing. In terms of somatic modifications, by contrast, enhancement possibilities are clearly more limited. Nonetheless, there may be options here as well. Treatments devised to alleviate muscular dystrophy symptoms might be used in healthy people to enhance muscle power. This would be an example of the so-called off-label use of medications licensed for medical use. This would be legal. You only have to find a collaborating medical practitioner. But that's not an essentially novel situation. We've had this with other doping methods. Now let's see. Let's look at enhancements from an ethical point of view. These enhancements are certainly a divisive topic in the general public. Of course, several potential arguments against enhancements come to mind immediately. According to a widespread critique, certain enhanced features would simply not be natural. And that's probably the gut reaction many people have. Upon reflection, however, it is not so easy to say what is natural in humans. But more importantly, it is not the case that what is natural, even if you can't flesh that out, is at the same time normative. The mere fact that by nature almost everybody does X does not mean that everybody should indeed do X. Indeed, this is not the way classical ethics have appealed to nature. Traditionally, nature has been understood to provide the conditions under which humans flourish. So, if humans wish to lead a good life, then the advice is that they better stick to the boundaries that are commonly found in nature. The difficulty is, however, that we cannot define a priori what circumstances people will find fulfilling. Certainly, I cannot imagine that anyone living for, say, over 150 years would find life genuinely more fulfilling than life under current conditions. But suppose I found a magic potion for just a little extra intelligence without other side effects. I don't think we can completely rule out that anyone would find life with a potion more fulfilling than without. So the concept of nature may be of limited use here. Another common sense objection against enhancements is that they may cement and increase social inequalities. Perhaps we should not tolerate any enhancements that lead to substantial inequality. At the same time, there might be genetic enhancements that privilege certain individuals in such a way, however, that others are left better off at the same time, even if the one enhanced person profits significantly more than others. Suppose that those who also profit are not just part of some exclusive clique, but that such wider profits are open and accessible. Traditionally, this has been the case with state-funded universities, where only certain individuals profit in the short-term perspective, but in the longer term, this privilege is distributed more widely, and it seems that the benefits on the whole increase. So I don't think we can so if a similar effect might be achieved with genetic enhancements, it would be difficult to object on purely egalitarian grounds. So if the argument of nature and of egalitarianism does not strictly rule out enhancements, how about the notion that we might play God? Here's somebody who's written about that question. Might we interfere with the Creator's work in choosing enhancements? Theologian Ted Peters suggests that small enhancements might help us lead more fulfilling lives, as long as there is no coercion and they do not increase social inequality. There are two theological arguments especially. To begin with, Peters sees creation in God's image as the call to assist God in creation. We are created co-creators. Think of how in Genesis 2, Adam looks for a peer among the other creatures. It seems almost to be to everybody's surprise that he does not find a partner. That really turns out to be the case. And only then does God proceed to create Eve. This at last is bone of my bones. Here the human person is intricately involved, an active participant in shaping the world of creatures. And it's possible to come up with several other examples from biblical traditions of this sort of dynamic. So plausibly enough, being a creature does not necessarily mean that we must simply respect what is the case by nature. After all, cultural achievements like communication or hunting in the evolution of humanity, or education, have changed what it is to be human over the course of millennia. We do in fact use many little enhancements every day in nutrition or technology, for example. Another one of Peters's arguments refers to the theological doctrine of ultimate salvation, to eschatology rather than creation. He sees science and technology as a human anticipation in God's eschatological transformation, a human anticipation of God's eschatological transformation that has already been at work in Jesus' healing ministry. Of course, we do not bring about eschatological salvation ourselves, Peters says. That is God's work alone, just as God is the creator and we are the creature. Yet, given that ultimate fulfillment is our destiny, Peters argues it is only appropriate that we foreshadow ultimate salvation already in this life, in claiming those possibilities that already in this life give us a vague glimpse of ultimate fulfillment. Peters further distinguishes between small-scale enhancements and transhumanism. We should indeed not allow for interventions that drastically change what it means to be human, he submits. It is important, however, to ask why he is against such transhumanism. He considers the suggestion that we might replace our current life form with intelligent computers to which we upload our personalities, which might then go on living forever, free of the 1,000 shocks that the human flesh is heir to. Peters calls this vision dubious and unrealistic, however. So on this side of God's ultimate salvation, the human task would be much more modest. Yet, what if we found ways of radical enhancement that are more realistic technologically, perhaps involving genetic modifications? Peters does not say, but I do not see why he would not be on board. But first of all, let's look at small genetic enhancements. To be sure, we should not disdain the criterion of human fulfillment, and physical integrity can play a significant role there, which resonates with Jesus' healing ministry. So Jesus did not just preach some ethereal gospel of the pure spirit. Why not go a little further then and boost flourishing with enhancements? It seems to me that Ted Peters' notion of cultural values and theological, it seems to me that Ted Peters' notion of anticipating ultimate redemption by increasing human fulfillment here and now short-circuits common cultural values and theological grounds for hope. He extrapolates from current standards to the way final salvation will be. True, he adds the caveat that divine redemption will not itself take the form that enhancements offer, but nonetheless enhancements seem to him a suitable parable of salvation or a correspondence to salvation. While Peters rightly views ultimate salvation as saving and ennobling or as perfecting, our theological vision, in my opinion, should also include the aspect of judgment that has traditionally been part of eschatological hope. So interestingly, in Jonathan's talk, there was also the aspect of judgment, which might undermine our current efforts to preserve nature, biodiversity. But I think it's not an either-or, either perfecting or judging, but both aspects should be present. And with judgment, I do not mean this radical notion of complete destruction that might be floated sometimes. And I do not mean the eternal damnation of sinners or non-believers. But what I'm thinking of is some painful process of separation from those things that keep us from being fully transformed into God's image. So taking leave of the flesh pots of Egypt can be a painful process. Such a transformation would not be credible if it did not involve some pain and difficulty. Discomfort of this sort you would envision human sinners to feel when they are liberated from the illusions that they hold so dear. In this sense, ultimate salvation, even for believing Christians, cannot simply be a maximization of those things we value in this life. In many accounts, it is indeed a dynamic of maximization that seems to shape the doctrine of ultimate redemption. For example, we sometimes find theologians equating God's eternity simply with infinity. A strong focus on the doctrine of creation, for example, might highlight the contrast between creature and creator and say that the contrast is one of finitude versus infinity. The doctrine of redemption, eschatology, is sometimes also phrased in such terms. In fact, to characterize God and ultimate salvation primarily as infinity might not say anything helpful if we wish to think of God in specific terms that also excludes something. So my suggestion instead would be to look also for those subversive notions of divine redemption that undermine the extension of this worldly progress and maximize temporal values the way that Peter sees enhancements doing. And my final point, what I have not spelled out yet is why we should, on the whole, be so critical of enhancements. Even if Ted Peters's eschatological argument might raise questions. Perhaps these enhancements do not anticipate eschatological redemption, but that does not necessarily mean that they are immoral. At this point, I wish to focus on enhancements via germline intervention. Parents might decide on enhancements that would not benefit themselves, but they hope their children. That's the thing, of course, with germline interventions. You cannot profit from them yourself directly, but your descendants would. Theologian Ronald Cole Turner emphasizes that we must not assume bad motives on the part of those parents that might choose genetic enhancements. Indeed, many parents want only the best for their children, and not in a narrow-minded competitive sense. But it remains a crucial point that children cannot consent to enhancements via germline modification. Sure, they cannot consent to medical procedures either, but there is reason to presume consent in that case of medical procedures. Enhancements, by contrast, are driven by particular personal preferences. Parents undergo IVF, pay significant amounts of money, say, to boost their child's academic achievement, and they undergo a procedure that is never entirely without risk. It is unlikely, in my opinion, that this child will not be prodded and pushed to do well at school and beyond. Yet, in spite of the genetic intervention, it is certainly possible that the child is more interested in, say, sports or music. So we're setting up a conflict, potentially, with those enhancements. And, of course, manipulative, ambitious parenting is a problem even without enhancements. To illustrate I have chosen a picture from a children's marathon held last year in Austria that was called off because parents were too ambitious, and here you can see them pulling their screaming children across the finish line. Now, it is, of course, unrealistic, one might object, for parents to let the child make all the important decisions herself. Parents do make various choices for the child unilaterally. And even there, we can't wait for the child's consent. And for the most part, that is fine. To highlight the problem of consent in the enhancement question does not mean that parents should not commit to schooling or religious education. But at school and in religious communities, we encourage approaches that are appropriate to the child's developing mind, rather than simply pressuring the child into compliance. By contrast, to choose enhancements is to choose a situation that is highly conducive to manipulation, And we should not justify that with the fact that parents make choices for the children anyway. That is my outlook on genetic modifications. Thank you.
Thank you very much, Alexander. Plenty of food for thought there, and I suspect there'll be lots of questions and comments. So over to you for the next 20 minutes or so.
Yes, can I just make a comment and then ask a question? And it's going back not to enhancement, but to the question of disease and also genome-wide association studies. You mentioned the one where they identified 74 loci in kids with high educational attainment or so forth. I think it's probably worth, just this as a comment really, just to highlight the point, just so people understand that those studies based on genome-wide association studies are population studies. And so hundreds of different genetic variants have been statistically associated with groups of children with high educational attainment or with high IQ. And, you know, as you well know, there's lots of studies in the literature. So I think from that perspective, in terms of those who wish to modify the genome to have children who are highly intelligent, is really fantasy, you know, because there are hundreds and hundreds of loci, hundreds of genetic variants that appear to contribute, as you'd expect, to the variation in the population to that particular trait. Okay, so in other words, those transhumanists and people who think that you could amplify the intelligence in that way are just, they don't understand the biology is what I want to say really. They have no idea really of what it's about. So, and they don't understand, I think, what genome-wide association studies actually tell us. So that's just a comment. My question actually is on your own thoughts. You mentioned that those who are handicapped in various ways do have a high level of satisfaction in life very often. They've adapted, you know, to their condition. What you didn't mention was that in many genetic diseases, many Mendelian diseases, these are children who die before the age of 10, often slowly and painfully, and they're not in those studies. You're not going to get those in those studies because many of them are highly ill and, you know, deep in bad pathology, which often kills them, you know, before the age of 8 or 10. So do you think in those circumstances then, and those are single gene disorders, many of them, hundreds of them, actually. So do you think that's more justified then to use gene editing in the germline in those circumstances where perhaps PGD may not be applicable?
Yes, thank you. So the comment on those genome-wide association studies, I think that highlights 2 points. On the one hand, it's a really high number of genes that are involved. But then one might still argue, well, that's even better if I want to do something about my kid. There are more points at which I can intervene to increase intelligence. But the other point then comes in that there's a systemic quality, presumably, to all this, with all of these many, many factors that come in. So they interact in ways in which the relation between these individual genetic loci plays a role, and you can't just scale one factor up and expect the whole phenomenon that you're trying to achieve also to increase. So it's a different logic that really obtains there. That's right. So that was the point of fleshing that out a little. Now those genetic diseases, that's right. I think there's a difficult line to navigate here. So on the one hand, physical integrity does play a role for human fulfillment according to the wishes of the Creator. And I think we can read the Gospels in that sense, that God isn't simply indifferent to this. And it's not just a matter of mind that ignores those physical issues that can be quite painful, that can also impinge even on the resources with which you make meaning in that situation. So I would say that in severe cases that would justify either PGD or perhaps in those rare cases where PGD would not be applicable, we could look into, in my opinion, into germline intervention. The point that I want to highlight is I don't see the urgency for that at this point. So, for example, I've looked at the German debate on this issue coming from Germany. There were high-level discussions in public on national ethics panels that sort of wiped away the issue whether PGD might replace germline modification, and it led to some great enthusiasm about germline modification, which again contributes to this technological momentum that would have real effects on public discourse and might suggest choices to us which would be forced choices, which we don't have to make. And then, of course, the question is, how do you distinguish between those very severe diseases and others that we might well live with? I found diabetes as one of the cases where I would say that is not a question where I would find PGD or germline intervention appropriate. Another question is, of course, and this goes right into this wider area of prenatal ethics, which we might hear more about later this afternoon. In what cases might, say, an abortion be justified? I am a bit worried that conditions like Down syndrome would be severely reduced with continuing technological progress. But at the same time, I'm hesitant to say that society should intervene and forbid in any simple way. So at the end of the day, it's the family that has to cope with certain burdens, and there should be a legitimate role for parents to make the call. We are able to do that. So the question then becomes, how do we enable families to cope better with such challenges? And there I think these new technological developments call for increased investments into public infrastructure that make life with disability a little easier, which would, in my opinion, also be a sign of continuing social solidarity with people with disability.
Thank you, but in an international context, who finally is able to make the decisions and the laws we need?
Who's making the laws? At the end of the day, the lawyers and the parliaments have a crucial role. So, for example, in the UK, so far the procedure has been that the HFEA, the Human Fertility and Embryology authorities, are the institution that regulates these things by law, that has been regulated in the 1990s with an act of parliament. And they, maintain an ethics panel that looks individually into these questions. And for example, if a family applies to do PGD, they might say that is well warranted, both legally and ethically, so we'll grant this license. Or there have been cases where they have refused. So in a sense, this political commonwealth decided to have fairly broad regulations and to leave the individual decisions to a panel of experts, which might be beyond social control at that point. So it's very difficult. So ethicists always like to look at individual cases. It is the individual people in their particular situations that are faced with challenges, and they look for things they can do. But then the law needs to have some specificity. So perhaps some ethics panels might be an important, helpful way to regulate this. So they would first of all see, are we within the bounds of law in this case? Is it even allowed to implant a modified embryo? So this would be something that would be up for Parliament to decide. And what might be helpful is then to keep a close eye who sits on those panels and demand reports. So certain societal groups should be represented. At the end of the day, that is a complicated process of societal compromise, I think.
Does that include religious representation on these ethics panels, or is that not?
Well, that is not for me to say. I think at the moment, the religious representatives are not represented at the HFEA panels. If questions are particularly divisive socially, it could make good sense to have religious ethicists on those panels, because of course religions do touch on these deep-seated feelings that move people, even if people aren't always able to spell them out in the right ethical terminology, etc. So one thing that you might want to think of is, in an age of worries about populism, Brexit and Trump, these things can stir very profound emotions that can be used for power purposes or just distort the ethical discussions out of proportions. So it's the question, how do we engender compromise here that really works for people in ways that are helpful and ethical?
To pick up on that one a little bit further, of course, we have fairly good legislation in this country and in Europe and the US, but there's no international legislation that could be held binding. And there's always the rogue state, and particularly the rogue scientist, with the particular ego that would be at stake over this. The first person to achieve something would be remembered. Better to be remembered for murder, they say, than not to be remembered at all. Is it what can be done internationally? And granted also that a lot of these procedures would be undetectable afterwards. Unlike a lot of the old think they leave marker genes there. A lot of the cheating in sport that goes on with drugs is detectable. Many of these enhancements would simply be switching one natural gene for another natural gene, and there would be no markers left.
Yeah, very important question. In an age where science has become really internationalized, and it's been China that's been pushing the agenda somewhat, with at least 3 prominent studies on modified embryos. And interestingly, those studies have not been published in very reputable journals. So that's the case of the game theory question: who's the defector that does the dubious thing first? Perhaps it is not dubious, we don't know, but this is speaking under particular premises. One author who also looks at the legal aspect, international regulation, is Francis Fukuyama, who wrote the book on our post-human future. And he is very much concerned about these procedures and wants to encourage people that we shouldn't just throw our hands in the air and say, oh, everything's so complex, there's nothing we can do. We have worked out certain regulations, say in nuclear proliferation, which doesn't work great, but it could work. And there's the question, of course, with international treaties and climate change. So there might be ways that international regulations could come in. What do we do with those interventions that we can't detect anymore? I guess we'll just live with those people who have been modified. The question is, will they have if they have been impaired by these procedures, will they have some way of regress there? So in China, these studies that have been done they were given the green light by university ethics boards. That is not a strong form of oversight. But at the same time, it's remarkable that it seems those modified embryos have not been implanted yet. So maybe not all is lost there. In sports, the question is then, there we should really detect who these people are. That is a huge question. One possibility might perhaps to require athletes to submit DNA probes from their parents as well. But who knows? Doping is a very complicated question.
Time for one last question.
With biobanks, I just wondered the storage of when people hand over this stuff for study. I wondered about the theological implications of this. So say I have my genome mapped, this isn't my specialism, and you store this. What if 10 years time down the road they want to use it for something else? Are there ethical implications that I can say I'm a Christian, I believe this, 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 and this, so in the future I wouldn't want this? Have they started to think about these theological implications of collecting all this stuff to then look into it?
Yeah, one question is, what does biobanks do, or the 100,000 Genomes project of the NHS? The good thing with those is that those are fairly well regulated, and they've thought carefully about how to handle this information responsibly. You have pretty strict requirements of informed consent there, and they can't just do anything. You don't write them a blank check. You can revoke your consent. You can also, if they find something new that they might look for, it's possible that they would approach you for a new consent form to sign. What I'm a bit more worried about is those direct-to-consumer testing agencies, 23andMe. So what happens with those data? That's the big question. On the other hand, I was at a panel recently where somebody who works closely with these projects raised the question, well, what are you afraid that might happen? Now, everybody has a right to privacy, and that's a right everybody has per se, whatever might come out of it. On the other hand, what are the accidents, what are the bad consequences that might really happen? It's not a foregone conclusion that, for example, insurance companies would be really greedy for more data and mine whatever they can get their hands on. That's not so clear, because maybe genetic information isn't so predictive as we think. If insurance companies really try to get all the data, they might run out of people they could insure. But again, it's a bit of a blurry area there, yes.
Thanks very much, Alexander. Let's thank him again.